Hello, everybody. So today we are going to discuss about our new topic, which is Bryophyta. Bryophyta basically were the first plants to evolve on land, and these plants basically are having characteristic that being the first plant on land, these were considered um, as the amphibians of the plant world. Amphibians of plant worlds, which means amphibians were the first animals which lived on land, first vertebrates which came on land. So these are amphibians of plant world because they have amphibious nature. What does this mean? This means that they are having water. They are dependent on water. They're dependent on water. They're dependent on water for their reproduction, which means the transfer of their male gametes, anthrozoids or spermatozoids, towards the female sex organ and archegonium is required to be done within a medium which is water, due to which that watery medium is found in wet areas either or near ponds or moist soil, as a result of which these are mostly found in those areas. Moreover, these are dependent on water such that their anthrozoids or spermatozoids are required to be dispersed. Anthrozoids or spermatozoids are required to be dispersed through water into the female sex organ archegonium within the medium of water. So in that case, water is essential for their reproduction, due to which they are important for they are considered, as a result of which as a result of which these are considered as as a result of which these are considered as amphibians of the plant world. And apart from that, these are also known as hydroscopic plants. Hydroscopic plants, reason behind it is that these are dependent on water, so these are known as hydroscopic plants, and they range from 1 to 20 centimeter, such that Markensia is the largest uh, bryophyta, which belongs to the subdivision Bryopsida, Markensia is the largest bryophyta which belongs to the subdivision Hepatici or Hepaticopsida. As a result of which, it is around 20 centimeter in length. It is the largest bryophyta. Moreover, when it comes to their characteristic, they have male sex organs. They have male sex organ which is known as antheridium. Of course, they have multicellular sex organ which is known as antheridium, in which there is production of the male gametes, which is antherozoites or spermozoites. Moreover, when it comes to the female sex organ, it involves female sex organ involves archegonium, which produces the egg. And now, when it comes to the female gamete, which is egg is basically it is immotile without flagella, immotile eye without flagella, haploid, single, larger in size, present within the structure of archegonium. While when it comes to antherozoid or spermozoid, these are motile, biflagellated, haploid, smaller in size, and multiple in number, right? So uh, these are multiple in number because they travel from antheridium towards the archegonium, due to which many of these are unable to survive within the environmental conditions, due to which there are more chances of their decaying, due to which they are produced large in number. While egg stays within the and archegonium, as a result of which it is only found single in number, right? And when the male gamete, sorry, so their body is not well differentiated, but it is differentiated into root-like structures known as rhizoids, and they have root-like structures rhizoids and stem-like structures pseudostems and leaf-like structures pseudoleaves, right? Apart from that, when it comes to their gametophyte, the gametophyte is a dominant generation. Gametophyte is a dominant generation. Reason behind it is that gametophyte basically has chlorophyll inside its chloroplast, due to which it is having photosynthesizing capability. Photosynthesizing capability is present within it. Sizing capability present within it due to presence of chlorophyll and into it, due to which they are able to synthesize their own food, as a result of which these are independent and not dependent on any other uh, plant. So these are independent, as a result of which when these are independent, they are more likely to live and have higher life expectancy as compared to sporophytes, as a result of which, as these are having higher life expectancy, these are considered as more dominant, considered as a dominant generation. While when it comes to sporophytes are semi-parasites. These are semi-parasites over semi-parasites. These are the semi-parasites on semi-parasites on the gametophytes, which means they are partially or completely dependent on gametophytes. Reason behind it is that they are not having chlorophyll. They are not having chlorophyll, and these are not having photos, which means if chlorophyll is not present, there is no maturation of chloroplast, due to which there is no photosynthesizing material present in them. So these do not have photosynthesizing capability, as a result of which these are dependent on gametophyte, as a result of which they have less life expectancy, which means after the death of gametophyte, they will be died as well. They will die as well, right? So in that case, they have less life expectancy, due to which these are conspicuous and less dominant. Conspicuous, less dominant, right? After that, these are homosporous, which means same morphologically similar spores are there, and these are usually found near wet rocks and soils, wet rocks and moist soils, and moist places as well. Reason behind it is that they require water for their reproduction, so moist areas are required in this condition. And apart from that, their male gametes are matured before their female gametes. Their male gametes mature before female gametes, and this process is known as protoentry. And the plant in which protoentry occurs is known as protoendria, which means bryophytes are protoendria, right? Protoendria. Oops. Proto Protoendria, right? So these are some common considerations. Apart from that, when it comes to some other characteristics, these are not having lignin. These are avascular, which means they are without any sort of vascular tissue. Xylem and phloem are absent in them, as well as they do not have lignin fortified tissues, which means lignin fortified tissues are absent in them, because these do not have any sort of upright structure, because of no hardened structures. These do not have lignin for fortifying tissues, because they do not have any sort of hardened structure, due to which they do not have lignin, as well as fortified. They do not have lignin, as well as they are avascular without uh, the vascular bundle, xylem and phloem, right? Similarly, there's another plant, pteridophyte. Pteridophyte basically are having pteridophyte are as bryophyte are having their gametophyte as their dominant generation. Pteridophyte are having sporophyte as their dominant generation. We are going to study that in our next class, right? So here, pteridophytes, sporophytes are dominant in the pteridophytes, as well as when it comes to the male and female gametes, antheridium and archegonium, these are found on the apex of the stem. Found on the apex of the stem. These are found on the apex of the stem. Sorry, not on the stem, apex of the branch, because they do not have a stem, right? And after that, when it comes to some other characteristics, the theta is basically we are having certain characteristics of the sporophyte when it comes to the anatomy of sporophyte. So sporophyte basically, it is a structure which is developed in such a manner that here we are having, you know, let's suppose this is a gametophyte. Let me just show you in the image. This is a gametophyte, let's suppose, and over that gametophyte there is development of a sporophyte, and its sporophyte is developed in such a manner that here there is arrival of a foot, or a stalk which is arisen from the gametophyte, right? So let me just try to draw a structure. Here we are having a gametophyte, let's suppose, and just it's a rough structure again. And over the gametophyte, there is development of a stalk of sporophyte, and that stalk of sporophyte which is attached to the gametophyte for absorption of nutrients from it and getting nutrition from it is known as foot, right? And that foot is having a structure, particularly a structure is present uh, within the foot which is known as the capsule, and that capsule is also known as sporogonium. And whenever there's asexual reproduction occurring, asexual reproduction occurs whenever there's development of the spores from the haploid gametes. There's diploid zygote produced, as a result of which embryo is produced, and that embryo, which undergoes repetitive cell division, first produces a structure of capsule. First produces a structure of capsule or sporogonium, and from this capsule or sporogonium there is development of foot, and then there is development of another structure, another branch like structure which is known as seta. Seta, it is also known as stalk. Seta, also known as stalk. And in some of the plants, some plants including let's just take example of Bryopsida. Bryopsida are basically having characteristics. There's Bryopsida are examples of this. And we are going to discuss the classification. We'll be discussing that in detail. That there are three major subdivisions: Bryopsida, Hepaticopsida or Hepatici, Bryopsida or Muscae, and the third subdivision which is involved in them is Anthoceropsida or Anthoceti. So here, when we are going to discuss Bryopsida, Bryopsida are having multicellular hair-like structures around the capsule in the sporophyte, which are known as paraphysis. Paraphysis. Paraphysis, which are known as paraphysis, right? This this structure is known as paraphysis. They are tiny hair-like structures, multicellular hair-like structures known as paraphysis, and these paraphysis help in the dispersal of spores during the asexual reproductive phase, right? And uh, here when it comes to some other characteristics, we are also having the anatomy of archegonium. If we talk about archegonium, archegonium is developed in such a manner that here we are having uh, a structure of archegonium which is known as neck. I'm just telling it from upside, that here there is a neck of the archegonium which is made up of several neck cells, and after neck we are having the structure, a capsule like structure which is known as venter. Within the venter we are having the egg, right? This is the egg present within the venter which is haploid, and the downwards the vector venter we are basically having a stalk, and this is known as venter, right? And here that venter contains the egg, where there is that venter contains the egg, which basically is where the fertilization occurs, i.e. the sperm enters within the archegonium into the venter, where it fuses with the egg, as a result of which it causes to produce a diploid zygote within the venter. Once the diploid zygote is produced, that diploid zygote now undergoes repetitive mitotic divisions, as a result of which it causes to produce a multicellular embryo within the venter, due to which the multicellular embryo is retained within the venter for a certain period of time. And after that resting period, that multicellular embryo is then developed into the structure of multicellular embryo is then developed into the structure of sporogonium or capsule by repetitive mitotic division occurring within it. as a result of which that sporogonium further causes to form rest of the structure of seta as well as foot from it in order to develop a separate sporophyte over the gametophyte. So in this case, this is how the development occurs. Moreover, when it comes to the traveling of spermozoids, this is, let's suppose this is a spermozoids. Let's suppose these are the two, what do we call it, flagella. Biflagellated sperm is there, which is motile, and this biflagellated sperm is attracted towards the archegonium by the secretion of a sugary substance from the archegonium, a sugary substance from the archegonium, a sugary chemical substance from the archegonium, as a result of which the biflagellated spermozoid or antherozoid is attracted towards the archegonium in order to enter into the archegonium into the venter and fuse with the egg. Therefore, this type of movement which is occurring over here is known as chemotactic movement, and this chemotactic movement leads to the fusion, right? After that, when it comes to their haploid life cycle, their haploid life cycle starts from their, when it comes to their haploid life cycle, that starts from the spore, haploid spores, and ends into the haploid gametes, whereas the diploid life cycle starts from the diploid zygote and ends into the spore mother cell. So these are some characteristics. Apart from which, when it comes to the classification, we are having three major subdivisions of the phylum Bryophyta, which includes Hepaticopsida, Hepatici. Number one, Hepaticopsida, Hepatici, in which we are having Mercantia, and second class, second subdivision which we are having is basically the Bryopsida or Muscae, and third is includes Anthoceropsida or Anthoceti. Now, when it comes to Hepaticopsida or Hepatici, it basically involves organisms. It involves around 900 species. Out of 300, there are around 360,000 species of plant in the kingdom Plantae, out of which 900 species are found in subdivision Hepatici, Hepaticopsida. And among that, these are organisms which inhabit wet areas such as wet land, wet rocks, as well as moist places where these are wet rocks, wet rocks, and wet rocks and wet places. Wet rocks or wet places. So these wet rocks or wet places are there from where they reside, and this is where they are, grown. So as they are grown on wet places of wet, moist places or wet rocks, they have less chances of getting, and they have less chances of getting desiccated, which means they have less chances of loss of water, due to which their loss of water is greatly reduced. And apart from that, they have developed a compact multicellular body which helps them to adapt themselves on plant. As plants, they have compact multicellular body which helps them to adapt on land, as well as um, they have a body which is, for, you know they have a simple body which is thalloid. They have a simple body which is thalloid with root-like differentiated structure, root-like structures known as rhizoids. They anchor their body, thalloid body, with they anchor their thalloid body with the surface rock. In case of organisms such as those which have thalloid body, they anchor their body with the surface rock with the help of root-like structures known as rhizoids. And examples of these organisms includes Markentia. Markentia involves this. This involves examples of Markentia. As I previously told, Markentia is involved in Hepaticopsida. So Markentia, which is 20 centimeter long, largest bryophyta, is basically having a thalloid formation with root-like structures known as the rhizoids, which allow them for their anchorage. to the rocks, as a result of which this is how the structure looks like. Here there's a diagram as well, a real life picture. This the picture you can see here. Over here, this is basically a picture of Markantia, and as you can see, these are 20 centimeter long, and these are the largest bryophyta, right? After that, after that, when it comes to after that, when it comes to some other structures, we are having other types of bryophyta as well. By the way, bryophyta are the simplest, the hepaticopsida are the simplest bryophyta, among all bryophyta. These are the simplest bryophyta. And hepaticopsis, other example includes porella. Porella is basically having rhizoids, but alongside that it is having its thyloid differentiated into pseudo stems and pseudo leaves as well. So this is one characteristics as well, right? And after that, the Markensia is also having its anthridium and archegonium developed on special branches, which is known as anthridiophores and archegoniophores. As you can see in the diagram as well, I might show you that here there is anthridiophore developed, and here there is archegoniophores, right? So anthridiophores and archegoniophores are developed as well. Here, anthridiophores is a smaller one, archegoniophore is a bit larger one. Archegoniophore is a bit larger one, right? And their life cycle is, almost similar to, other bryophyta as well. So we are going to discuss in as a single example of life cycle of most funaria, right? After that, when it comes to some other structures, there's Bryopsida muscae. Bryopsida muscae basically involves the example of funaria. By the way, these can be monoecious as well as dioecious. When it comes to Bryopsida are also having, they are a bit advanced than Hepaticopsida, such that they are having, you know, the following structures except Funaria. Funaria basically it is monoecious, which means uh, it is monoecious. Funaria is monoecious. Funaria is monoecious, and Funaria is monoecious, which means it is having its male and female gametes, antheridium, and archegonium. It has its antheridium and archegonium developed on the same plant on different branches. Whereas when it comes to some of dioecious example, there's polytrichum. Polytrichum is having its antheridium and archegonium developed on different plants on different branches, right? And apart from that, these are also, they have a body. One more thing I forgot to mention in the case of Bryopsida. Bryopsida have a body which is basically having, it is dichotomously branched, dichotomous body. Dichotomously branched, which means they have a body which has a main axis, and from that main axis there is development of two branches, which is known as dichotomously branched body, right? And apart from that, when it comes to Bryopsida, they are having, a polytrichum as the dioecious ones, as well as their reproduction is almost same, except the fact that when the spores are developed, the haploid spores are grown into a filamentous. When the haploid spores are developed, dispersed, these haploid spores developed these haploid spores are developed. Let me just write it over here. Haploid spores are developed into a filamentous alga-like structure which is known as protonema. And that protonema has the characteristic that it is having several branches, and over those branches there's development of a bud. There are several, branches, like filamentous branches are there, alga-like filamentous branches, and over those branches there are buds developed. There's development of several buds. There is development of several buds, and from that individual buds there grows an individual gametophyte. There's development of an individual gametophyte, which basically causes the degradation of the protonema once a gametophyte is developed. Once the gametophyte is developed, the protonema is degraded and disappeared once the gametophyte is developed, right? And this is what we consider in case of the Bryopsida, right? After that, when it comes to some other characteristics, we also having their picture over here. As you can see in the diagram, there is main axis, which is divided into two major branches, which is known as dichotomous, and rhizoids are also present in them, differentiated their roots and stems. There's no stem and leaves like structure, so they only have pseudo stems and pseudo leaves. Pseudostems and pseudoleaves. Moreover, they have a seta, which is developed over the gametophyte as a semi-parasite like structure. Apart from which, when it comes to some other characteristics, the Bryopsida are also having they are having paraphysis. Bryopsida are having paraphysis, but Hepaticopsida are not having paraphysis. This is the key point, exam MCQ's point. Apart from this, when it comes to Ethnoceropsida, these are basically advanced version. These are advanced. Then Ethnoceropsida are advanced. Then Hepaticopsida and Bryopsida, such that they have sporophyte which is not dependent on gametophyte only in the mature stage. In the mature stage it is not dependent on the gametophyte, but in the young stage it is dependent on gametophyte due to no formation of chlorophyll within it, due to which it does not has any photosynthesizing capability. When it comes to develop structure of gametophyte is having characteristics such as it is highly lobed, it is irregular in outline, as well as it is highly lobed, irregular in outline. It is highly low. It is irregular in outline, as well as the Anthridium and Archegonium are partially submerged. Anthridium and Archegonium are partially submerged into the gametophytic tissues, as well as they have executin, which prevents desiccation and filters, a substantial amount of the ultraviolet rays from the sunlight. When it comes to the sporophytes, sporophytes are well developed such that they have chlorophyll in the epidermis. So they have photosynthesizing capability. They have stomata in the epidermis, which means they can diffuse gases such as carbon dioxide and oxygen, as well as they have uh, vaxicutin to prevent desiccation, as well as ultraviolet to prevent them from ultraviolet radiations from sunlight, as well as they as you can see in the diagram as well, this is example of an Anthoceropsida, which is also found in the hilly areas of Pakistan, known as Anthoceros, right? Anthoceros found in hilly areas of Pakistan. And one more thing that when it comes to the structure of their when it comes to the structure of the sporophyte has a ring. Sporophyte has basically a ring or band of meristematic tissues, a ring or band of meristematic tissues which is present between the foot and the spore producing region of the capsule or sporogonium, due to which that meristematic tissue ring basically causes to add cells on the spore producing region, causes to add cells over meristematic cells over the spore producing region of the capsule or sporogonium, which helps in the maturation, formation, and the dispersal of spores from the spore producing region. Moreover, this meristematic tissue basically has a high growth rate, fast growth rate, due to which it is it has high growth rate, fast growth rate. It has a high growth rate. It has a fast growth rate, due to which it continually increases its length, with an indefinite period of time, due to which even after the death and decay of the gametophyte, they are able to survive into a new condition. They, even though after the death and decay of the gametophyte, they are able to survive after the death and decay of gametophyte because of the growth, continual growth occurring due to their ring of meristematic tissues, as a result of which they are considered to be less conspicuous and more dominant in this condition. And one more thing that I would like to highlight over here is about um, is about uh, their conditions, that where do they live. They can live in fairly dry places because they have land adaptations, so they can fairly live in dry places. Moreover, when it comes to life cycle of moss, we are going to quickly take um, although many of the things are previously described in the video, but we are just going to take a quick look up into it. That's here. The life cycle starts with the dominant stage here, as we are having the gametophyte. So gametophyte basically has several archegonium and antheridium developed on its branches, and it is basically having the neck canal cells, over which there's venter, in which there's egg, and from that and on the other side we are having the antheridium, which is having the opercular cells, from where the sperm cells are transferred through the water medium, through chemotactic movement towards the archegonium, where it gets fused with the egg, due to which it causes to produce a zygote. Now that zygote produced is also known as oospore, diploid zygote, which is known as oospore, and that oospore develops into a two-celled zygote, epibasal, hypobasal, and that two-celled zygote is now further developed into epicell cell and basal cell, due to which that undergoes repetitive division of mitosis in order to produce embryo, from where that embryo undergoes multiple repetitive mitotic divisions, as a result of which it causes to produce a structure of the sporophyte over that gametophyte, in which there is seta, capsule, as well as the development of foot or calyptra. Moreover, a foot is also known as calyptra, by the way, right? After that, when it comes to the sporophyte has development of spore mother cell. That the spore mother cell undergoes meiosis, as a result of which it causes to produce a spore tetrad, which means four haploid spores are produced. Tetrad refers to the four haploid spores, due to which each haploid spore is then further germinated into a germ tube. Germination of a spore occurs into a germ tube, and from the germ tube there is development of filamentous body. There's development of a filamentous alga-like structure, which is known as the protonema, and that protonema further causes to produce several branches over which there are buds, and these buds ultimately grow into a separate gametophyte by the mitotic division, as a result of which the protonema which is developed is now degenerated or disappeared once the gametophyte are developed. So this is how the life cycle occurs. And gametophyte is the gamete producing generation, sporophyte is the spore producing generation. The haploid life cycle starts from the haploid spores and ends at the haploid gametes. Moreover, the diploid life cycle or the diploid life cycle or diploid generation starts from the diploid zygote or oospore and ends at the spore mother cell, which is diploid. So this is all about this video. Hopefully we'll meet again in another video.